Hi everyone, Cameron Hamilton here, Director of Planning at Ballast, and I'm here in conversation this week with Dr. Maj-Linda Selenica, and she's joining us to take us back to school this week. It's a little bit different than some of the conversations we normally have, but this is kind of a conversation I think that is sprouted from a passion I have, which is helping people that are affected by Alzheimer's. It's something that I've had in my family, and some of our clients know I helped start a nonprofit 15 years ago called Bike4Alz, where the college students ride across the country each year, and they just finished that this year for the 14th time. And through that, I have found partners and friends at the Sanders Brown Center on Aging here. So in Lexington, Kentucky, we have one of 30 centers on aging that is recognized by the NIH. Maybe you'll tell me better or correct me about that.
No, you're good.
Really a world-class center with world-class talent, including Maj-Linda, who has roots in Sweden. Is that
Yes,
correct?
That's correct.
So I think people would be interested to learn how you find your way to Lexington, Kentucky to make a career through this vocation you have.
Yeah, it's fantastic to be here. A pleasure really to talk about this with you and your clients and people in community. But I have a PhD in neuroscience, and that kind of directed the research through Alzheimer's disease. I really moved from Sweden to America because I wanted to do a fellowship. It's called the postdoctoral fellowship, which was in Florida, Tampa. And then going down kind of the career road for research and assistant professorship. But then when Linda Van Eldik, which is the director of our Sanders Brown Center on Aging here in Kentucky, had an interest in my research and our research that we do, and invited us to come to Kentucky, that was kind of an easy choice because, as you mentioned, Sanders Brown is really and truly one of the best centers in Alzheimer's disease research, but also leads a lot of initiatives in clinical trials, and I'm going to talk a bit about it as well when it comes to Alzheimer's, and has evolved with time and has recruited the best minds. They have clinicians that are the best in the country internationally, but also the researchers, and we're evolving with time of where the research is on Alzheimer's disease. They felt absolutely kind of where you want to be if you want to really cure and work towards Alzheimer's disease.
Right. So it's not just the basketball team here that recruits the top talent.
Unfortunately, it was not the basketball team.
Well, I'm saying, like the best players, we get the best Alzheimer's researchers here.
Yes. Absolutely, and that's a strive for sure.
Fantastic. So you have a presentation for us today about Alzheimer's and dementia and the work that you and your colleagues do at Sanders Brown.
Yeah, and I'm going to talk a bit about it, but again, this is kind of a big kind of project trying to introduce what Alzheimer's is and what we do in the field, what my own laboratory is doing, and how that fits within the Sanders Brown kind of vision. So I'm going to try to run through those concepts a little bit and touch upon them, and happy to stop for questions from people that potentially watch this webinar and have further questions to you. Please, happy to talk more about it.
Well, I think there's one of us in here that has passed biochemistry, so I always say, speak as if you're speaking to a small child, and I'm sure if you do that, everyone will appreciate what you have to say.
All right, well, we'll get it started. So Maj-Linda Selenica, Associate Professor at Sanders Brown. The title really is Alzheimer's disease and dementia, and then what we do is research from bench to studies and therapeutic strategies. So I just wanted to present the lab because really this is a team effort, as everything else that we do in our field. It's a laboratory full of very excited students, from undergrads to PhD students and postdoctoral, and then funding from NIH, but also funding. This is actually a picture with a check that we got from Bike4Alz, which
there we go.
Cameron said we've been fortunate enough, my laboratory, to actually been donated money from that enormous endeavor. I'm so impressed with this young guy.
Well, I'm the treasurer now, and they've raised $100,000 this year to date. So there's another check coming to Sanders Brown, and how I met you, I didn't say earlier, was when we went to Dr. Van Eldik and the team and said, we really want to make as big of an impact as we can with this relatively small amount of dollars. We're not raising $1 million each year. Can you take us to the PIs, the principal investigators, and say what could help them be more effective? And you were one of the scientists that stepped up and said, maybe if we have a piece of equipment, we can get a little bit of leverage out of that. So we were really excited to meet you and make that happen.
I mean, thank you for that. And we still use it. It helps us in so many different ways. That is a conversation for another time. But so the laboratory really focuses then, and we approach this a bit differently potentially from many laboratories in the country, but we do look at mechanisms that are involved in Alzheimer's disease. We want to cure it, and to cure the disease, you have to understand what's causing it at that molecular level, and we really look at it at that level when we investigate not only one thing that goes wrong, but potentially 2 or 3 things that go wrong.
Sure.
And why we do so, and I'm going to talk again a bit more details, is because we know that it's a complex disease where many things go wrong. Mm-hmm. So we have moved as a field, and definitely my laboratory has understood that for a long time now, that we have to tackle it as what we call a co-pathology here. And then the ultimate effort is obviously to kind of develop therapeutic strategies for Alzheimer's disease. Again, when we're talking about research, that is potentially in a small plate, in small animal models, but that is what goes then towards clinical trials and potentially an FDA-approved cure.
Yeah, that's everyone's goal in science, it seems, is let's go from the bench, which is me staring over a microscope, to the bedside, right, and translate it, and I think that's the goal at Sanders Brown.
That's absolutely the goal, and that's how we approach the whole research in Sanders Brown. Is what we call translational research. It has to apply to the human in a bedside. But kind of stepping back with Alzheimer's diseases, and I know that we talk in broad terms, and everybody thinks it's dementia, but we have to remember that it's really just one type of dementia, and very important to understand that. Is obviously the most common form of dementia. If you look at the numbers, which really this comes from Alzheimer's Association, that is 60 to 80% of dementia cases are most probably Alzheimer's disease, but really is just one of the big terms. Under this big umbrella of Alzheimer's disease, then we also have vascular dementia. We have Lewy body dementia with Parkinson, and I know a lot of people do recognize Parkinson as a dementia form, the frontal temporal dementia. So there's many breakdowns of this dementia, and within that little sleeve of frontal temporal dementia that has said 10 to 15%, there you have 20 different diseases.
Right.
So you understand how complex each sleeve of this dementia umbrella really is. My laboratory focuses in several of these forms, from Alzheimer's to frontal temporal dementia, so does Sanders Brown. As we know, because we all kind of involved or been impacted by this in certain levels, the patients with Alzheimer's disease express a variety of issues that we recognize. Obviously, forgetfulness, right? Not remembering, and memory loss is the most common one that we recognize. But then it's depression and confusion and disability and anxiety. So many symptoms, behavior symptoms that are associated with that. Language problems, I think it's a big one that we recognize, that's overall cognitive impairment, as it's called. But then we kind of tend not to talk much about that this is a family problem, because you have the caregivers that are impacted largely by it. So having really a family support system is very important. So we are affected by this disease in different social economic levels. And I do write some of the symptoms that are obviously recognized by the clinicians and medical community, and people can read into it, but is this communication problem and difficulty walking and speaking and swallowing. Those are kind of, as the disease progresses, you develop those different issues. So I like to put this slide up because for many reasons, but Alzheimer's disease was recognized and discovered by Dr. Alois Alzheimer's in 1901, and I did the math.
There we go.
So we have 125 years of Alzheimer's disease, and that is kind of fact number one in a way. But what I'm going to talk through this is that a lot of the symptoms that we still research and still don't know and don't have the cure for is the same things that Dr. Alois Alzheimer's really discovered.
Right.
Auguste Deter is the classical patient of his. That's where the disease was really discovered. She was a young lady. I think she was 55 when she died. And she kind of, Alzheimer's, Alois not only describes her symptoms where she was alive, when she shows signs of illness, delusional thoughts, but then when she died at 55, he actually was a neuropathologist that looks at her brain and start truly drawing, he and his trainee, what Alzheimer's disease looked like in her brain. And this is what it looked then, 125 years ago, and this is what it looks today. And we talk about Alzheimer's disease as two hallmarks of the pathology in the brain.
Right.
And we're going to talk a lot about the pathology in the brain, so I'll take a minute here. This is a cartoon over here kind of showing your neurons dying, which is comprised of a protein that we're going to call tau, which I'm going to talk a bit about it, but it's more a neuronal protein. When it gets pathological, in a sense, neurons die. It's
Right.
very toxic. The most common, or actually I'm not even going to say most common, what defines Alzheimer's disease is actually presence of the structures over here that are called plaques. We also have it here. Those are things that are outside the neurons, so kind of lie around in the brain, but form these very sticky plaques, just as it sounds. And those two have been studied for 125 years, trying to understand its role from being very bad for you to what else is associated with this plaque. So these two kind of features of the pathology still very much studied, still pretty much so much more to discover and understand what
Right.
they do.
And so this young woman 125 years ago had early onset Alzheimer's, and these bad substances stacked up so quickly that it lights up like a Christmas tree
Yeah.
on old technology.
Yeah.
But now we're having the ability to notice small molecules at a much more sensitive level and across a lot of different types of patients, which is how we know about all the different types of diseases that might actually
Yeah.
arise differently.
And you have passed your biochemistry because that is my next slide, by the way. But yes, we still define Alzheimer's disease.
Right.
These two tombstones of the pathology that develop with time. And to your point, the plaques, right? We just saw the picture from 125 years ago. This is that we just published 2 years ago in Nature, showing how these plaques are not as simple as we think they are. All it says is there's so many different structures of these things that are attached to this neuron that you see here that can form all these different forms of the same molecule, as
Right.
to speak. So how do you attack this therapeutically is the question.
Right.
Because we thought we're just going to attack amyloid plaque, but we're learning, and this is the latest of the latest technologies called cryo-EM, for whoever's interested, that can discover this structure. So now we have to go and rethink, how do we target this structure and this structure and this structure and
Right.
this structure in order to have elimination of these plaques. And then as a field, you also want to question, well, is that enough? And that brings me to the next one, and this is understanding. I did mention a little bit, tau is the other protein, right? We have
Right.
plaques and tangles, and tau filaments is another protein that we invested as a field very much in early 2000s. Similar here too, it forms different structures and different filaments. I mentioned earlier that frontal temporal dementia is actually compromises of 20 different small diseases that have this protein. So we have to be very careful also when we diagnose this patient. Is it frontal temporal dementia or is it Alzheimer's disease?
Right.
And the therapies that we have available now actually could potentially make a difference. So this personalized medicine that we talk very much at Sanders Brown is also kind of very interesting in this concept, understanding how diseases can be different.
And are we aware of a positive purpose for tau that is needed prior to it becoming at a high concentration unhelpful? You know, so stabilizing protein, like neurons need tau, but we have too much here is the issue?
Okay, so yes, absolutely yes.
Yeah.
And there is actually some good news that I brought for this presentation today. It's both the protein itself is too much, and then it's what we call post-translation modifications. These proteins get kind of sticky knots on them, and if that occurs too much, then they stop doing. So tau, let's take this example, actually binds to a protein itself, and it's almost like something that drives through the highway, a wheel of a car, if you might. But if this modification occurs to tau, it is not allowed to attach to this car. The wheel comes off the car pretty much, so now you have breakdown of the neuron. And
Right.
sometimes it's as simple as that. So it's many processes. Just out of curiosity, I think it's very well to understand how these pathologies affect the brain. This is a normal brain. This is an Alzheimer's disease brain. If you put them on top of each other, you actually see how much the brain shrinks, and that shrinkage of the brain is truly neuronal death in the brain. So you have this shrinkage of the brain, and then certain other regions of the brain becomes enlarged, and they don't also, because they do that, they do not operate at what they're supposed to do. So it's many features that we can measure in a patient to show that it's Alzheimer's disease, and certain of these regions become affected, as we know as a field. So that's where we focus on. We image the brain to see what changed, and we know what we're looking for if we're going to diagnose for Alzheimer's disease. But you did ask, and the numbers, I don't know how much we want to focus on that, but two things that I wanted to bring up is that the main risk factor for Alzheimer's disease is aging. It's a disease of aging, and as our populations get healthier and we want to progress towards that healthy aging, that also increases the risk for Alzheimer's disease. So we have 74% of people age 75 and above get Alzheimer's. The numbers total is 7.2 million, and this is in America only, not global. So again, there is a age-related risk factor with Alzheimer's disease that we cannot escape.
Right.
And it brings that burden to, as I mentioned before, about the socioeconomic burden into both medical system, the families, and then also.
Oh, it's a public health crisis.
It's absolutely a public health crisis.
It completely wrecks a family if we don't have someone sitting here ready to help.
Yeah.
It's very expensive to care for someone. If we talk about, clients who might have independent living at roughly $5,000 in some areas, it might be $15,000 a month to care for someone if you don't have a caregiver, and if it's not there, if it's with a family member, someone's underemployed and not contributing. And you said something about getting healthier. The way I talk to people about it is we've done such a good job curing different types of cancers and having healthier practices and fewer people smoking, and a lot of those people are getting older, but unfortunately those are more people who can wind up with dementia.
Exactly. Other system fails.
So this work is increasingly important, in my opinion.
Yeah, thanks. Absolutely. But then, and I'm going to jump over this is really the breakdown by the age. But then what also gets expensive is as those new therapies that we have come up with, then it has to be picked up by Medicare and other insurance companies to allow for us to use. What is interesting here, and I think it gives people a perspective also, is how those different things fail in Alzheimer's disease brain. And what we have here from normal to abnormal, and Alzheimer's disease normally comes in phases that are either preclinical, and what happens there is these plaques that I mentioned start developing first, and then brain activity goes down. Then you enter a phase that is called mild cognitive impairment, when we potentially even might start noticing something in our loved one, right? Its brain activity goes down. What we cannot notice is this tau protein starts accumulating, right? And then as you enter full stages of dementia, you have your brain kind of shrinking, as I mentioned, that full-blown loss in memory that we kind of see. So there are different stages, and we have ultimately what we want is strategies that actually target early stages rather than late stages. But I am one of the believer when we actually could also treat the late stages if we are failing in kind of treating the early ones.
Well, that chart really speaks to my numbers mentality, and I like the curves, because the idea is in any of these therapies we're trying to do, we're just trying to shift that all to the right.
Yeah.
So
delay everything.
Exactly.
Until we find a cure. So one of the most exciting things that happen in Alzheimer's disease is actually the biomarker field. So this is pretty much in fluids, preferentially blood, not CSF, not the blood, not the spinal cord fluid, right? We do want plasma and blood-based. We didn't have that. And I want to claim as late as 5 years ago. So the field has evolved so much when we actually now have biomarkers to truly say that this is Alzheimer's disease. Before it used to be that it's dementia, rightly so, people said it's dementia, and it was after post-mortem, right, after the patient deceased, that you could go and say, oh yeah, it's Alzheimer's.
Right.
So these biomarkers allow us to actually say that it's Alzheimer's, and what we look at is amyloid beta, again, those plaques, tau, the protein that is in neurons, neurodegeneration, so basically the N stands for it, but it's how much neurons are dying, and then cognition, the MSC test, the clock, those tests that patients do with their clinician. And what it allows us is to measure it both in plasma and spinal cord fluids, both the amyloid levels and the tau levels, but also imaging. You can actually see if these parts of the brain are shining red, tells you that you have accumulation of these proteins in the brain. You don't want that. But again, it allows you to early see that this is happening, and then being enrolled in clinical trials, trying some of the therapeutics that are out there already kind of being tested. So this is a fantastic both imaging biomarker, but also measuring in the plasma biomarker.
Well, can I tell you a recent story that I heard about a cognitive test?
Please.
My grandfather turns 100 in September. And he was having a cognitive test, and I think it had been going on for about an hour, and he was a little bit worn down mentally. And they said, okay, can you write us a complete sentence? And he wrote, "I wish you would leave now," and handed it over. So that's how he passed his test.
Fantastic. I think, seriously, if they were to give me those tests, I'm most probably going to fail now and then. But it is, some of them are very functional. One is that you draw the clock and you see the decline of the patient just by how they write the numbers and where those numbers are located in the clock. So it actually does work, but very funny grandfather. So these are really, really good improvements. This is kind of that imaging that I was talking about. These are called PET scans, when you actually can see where it accumulates, but you can also measure, and I'm not going to go into the whole glucose and sugar levels in the brain, but the brain is the main organ that utilizes 20% of your glucose. Whatever you eat, 20% of it goes to the brain because you have that high brain activity.
Yeah.
Right? What happens with Alzheimer's disease and many other diseases is that we don't utilize it anymore. Our neurons lose the ability to utilize it. So we can measure that nowadays as well. So these are fantastic markers, imaging markers towards the diagnosis of Alzheimer's disease.
And so those are PET scans of living patients who might be in a longitudinal studies. They might be Kentucky residents who are coming in voluntarily, who we don't know whether they're going to have dementia or not, and we can follow them and get this type of data.
Yeah, and I mean, these are costly tests. They have to be covered, so I think in some sense you have to know that they are progressing towards Alzheimer's disease. This is for tau, for example. This developed 10 years ago only, and it was a great success when we got this. But also with amyloid beta, those are infusions, so there is a bit more complicated to get this. But definitely very much successful to do so. And then the next success story for me is that actually in April 2024, just 2 years ago, we had a different Alzheimer's blood biomarker test on the market. And if you look at these companies, those are all companies that we use for any other blood test that we do, right? So this is very, very encouraging that at least we can diagnose it early. We know if they are kind of pre or early Alzheimer's disease, we can start enrolling them in clinical trials and delay. And this is another in May 2025 that is this first FDA-approved test for diagnosis, because this was just the first set of tests was just to say, yes, you have increase in those proteins, but you've not been allowed to diagnose it. The one in 2025, and I put it down here, those are the levels of tau and amyloid beta. These actually allow then the clinician to diagnose for Alzheimer's disease. So both of them are great achievements in the field that we just didn't have them. Look at this, April 2024.
Yeah.
Amazing to know at how much we have actually evolved in the field. So what are some of the current treatments? I know it's great interest out there, and I think this is super important to mention. If you look at this table here from 1996 to 2014, and I remember when this drug came out because I was working at that company, we really just had these drugs that were, some of them from mild dementia to moderate and then severe, but all they did was to modify neuronal function. And they're okay, but they definitely did not delay the symptom. With time, the patients would kind of, you always adjust a little bit and then go back to where you were. So they were not success stories, but they were helpful, better than nothing. But then in 2020, we actually had this drug that we implemented for Alzheimer's disease when we tried to deal with the insomnia and all the sleep problems that that the patients had. And I think I have an animation, but if not, we'll stay. It's okay. Okay, there you go. In 2023 and 2024, we actually got 2 amyloid beta vaccines. And I think people have heard about it. That was the biggest breakthrough we had in the field to actually go what we call symptomatic, disease-modifying treatments. So the first set was symptomatic. You just dealing with symptoms of forgetting. This is actually going to exactly what the mechanisms of this are.
I recognize the name lecanemab.
Yeah,
Did Sanders Brown contribute to that?
Absolutely.
Yeah.
So Sanders Brown was one of the first sites that NIH FDA actually allowed to do the clinical trials, and we still have people enrolling for this drug. Donanemab is another vaccine that was approved. They, talking about the different structures, they actually target different structures of that amyloid plaque, again, because it's never enough with one of it, because so many different species in the brain that you need to tackle, and they have shown to be successful and kind of delaying this cognitive decline by 25%, so that is great news. I just happened to just come from Alzheimer's Association conference a week ago. That was in London, and hence Big Ben here.
There we go.
But I mean, things that excite me and excited the field the most is that now we have a clinical trial phase 3 that actually showed to be incredibly unprecedented successful at reducing this tau protein.
Wow.
You kind of hit it on a head there, asking, is it too much protein? And that's what treatment does. This really targets the protein itself, forget all the modifications of the protein, reduces as this protein builds up in the neurons, it reduces it. People that are interested in graphs, they can look at all it tells you is doses that reduce this protein, both in CSF and plasma, but also reduce the cognitive impairment. So basically make the patients up to 30% better.
Yeah.
And this is enormous. They have a dose now that they can go with and treat more patients. So this is an enormous success. The first time ever that a tau therapy is succeeded. And this other treatment also gets me, this is another therapy, vaccine therapy, makes me very excited for several reasons. Reason number one is that this one too affects amyloid, the plaques. And in fact, this antibody affects truly the plaque. It's shown extreme good success in reducing the plaques. More importantly, does not have ARIA, and this is kind of having those microbleeds in the brain, and a lot of immunotherapies could have those problems. This one had very low incidence of creating these microbleeds in the brain. So great success story. But what gets me excited is because they started 2 other trials globally implemented. With one, they're going to treat cognitively unimpaired individuals with high risk for AD. Can you imagine if that was actually a possibility? And then the other one, that they are going to have 2 global studies evaluating this treatment in early symptomatic ADs. So again, great therapies, great success stories in clinical trials for Alzheimer's disease that just came out of this. And this come a little bit my own research and kind of what we believe also in Sanders Brown and in the field of Alzheimer's disease. Alzheimer's disease is a mixed dementia. We learned the last 20 plus years that you don't have just this plaque and tau that I kept talking about, but you have alpha-synuclein, which most of people recognize as part of Parkinson. You know what? We find that in Alzheimer's disease brains. And we have this TDP-43 protein here that my laboratory very much focused on that is also part of Alzheimer's disease. So how do we tackle this, right? The picture suddenly becomes more complicated how do we tackle this?
Right.
All right. So now we're moving from Alzheimer's disease to what we call Alzheimer's disease related dementias, because now you have more of this. And we started by saying that in Sanders Brown, we really try to do translational research, which means that we do it in our systems in basic research levels, but then we go back to the bedside and make sure that that correlates with the patients do, and whatever we learn in our patient cohorts, we go back to the basic bench and try to understand those mechanisms. In our laboratory, the idea is that whatever we look into the basic research, that as implementation into the patient.
Right.
So it mimics what the patients are seeing, rather being cognition or being the brain pathology, right? And then we learn, because we have this brain bank and we have neuropathologist, Dr. Pete Nelson, that has been a fantastic scientist and helped our own research so much. When we find those findings in the patient brain, then we want to go back to our research and say, how do we modify our research questions that truly reflect what happens in a patient?
Yeah. Yeah, and you said we have this brain bank, and I want to highlight that because this is what I've been impressed with as I've come to learn about Sanders Brown is we've had for years and years a large cohort of people we've been studying who have donated their brains after they pass, and I think they said like if someone passes in the study, they have that brain sectioned like in a matter of hours.
In a matter of hours.
And we have the biggest tissue collection in the world.
So,
or one of the leading.
Yeah. No, so Dr. Pete Nelson is actually a neuropathologist, and he's taken over what Dr. Marksbury really built the whole Sanders Brown about being and having, that is having the community. We have the largest community-based brain bank. So what it means is that not only, and Dr. Marksbury actually came with that philosophy, in order for us to understand the disease, we need to understand what a healthy aging brain looks like.
Sure.
So he started collecting brains that were of aging population. So we have this control group to compare to, and it makes complete sense, right? So we have one of the largest brain banks when it comes to health, community healthy brain bank. And that allows us to have this translational research, which then ultimately obviously direct our clinical trials, how we go in a therapy about it.
Right. And I know we have clients here at Ballast that are part of that study.
Oh, wow. Fantastic. And we greatly appreciate it. Huge respect for doing that. So the research then in Sanders Brown really has several angles and several arms. Again, we're talking about mixed pathology. Our mission and vision is also healthy brain aging, so we want to understand the aging effects in a healthy individual. But then we also look at risk factors such as Down syndrome, vascular comorbidities, stroke and TBIs and all those things, inflammation, genetic, and lifestyle. And the idea is to kind of go from normal to all the stages of dementia, understanding where the clinical trial interventions should be.
Right.
So that brings me a little bit to my lab, because I mentioned at the beginning that we really approach this as we want to look at the mixed dementia angle of Alzheimer's disease. We are very good at understanding tau, but then this new protein called TDP43 was actually being discovered in early 2000, and this is the very young people in my lab that help and do all the work, so I have to acknowledge them. But actually, back to our brain bank and Dr. Pete Nelson, he actually published in 2019 this paper showing that 1 in 3 individuals that are over age 85, they develop a disease that has a very long name, but its short is late. So basically in their brains, they have this TDP43 pathology. And why is this important? Because when they did the math, they actually found that this late disease, this pathology, this TDP43, in rank, comes the third after Alzheimer's disease and vascular pathology. It's actually even before Parkinson and Lewy body, right? So huge public health risk, and nobody is looking at it. This protein normally is also associated with frontal temporal dementia and ALS, so it was a big effort in there, but nobody recognized that this is part of aging associated with Alzheimer's disease. Our lab investigated this a bit differently. I'm going to take some examples how I do it, but the other important thing to mention here is that and this is again where come out of Sanders Brown and Erin Abner and our center, did this looking at postmortem brains and did this post calculations and showed that for different diseases, every time that you had this TDP-43 in the brain, your cognition just got a bit worse and worse and worse. So basically it correlates better, or it drives that cognitive decline that we didn't know before. This is tau, and we thought tau is really bad. Really kills the neurons and declines the cognition. This is TDP-43 alone. Just in itself, this is dramatic. And you see that these proteins accumulate there. Your cognition just gets worse. So again, we understood that this protein is very much of interest. So how do we study? This is some case studies on my own research to show kind of how we apply those clinical findings actually in a bench side. And we use from animal models to this kind of what's going on in a cell behind the scenes and mechanisms. And the whole idea is to kind of find new ways to treat this co-pathologies, this mixed dementia phenotype that we see in the patients. So one thing that we do very well in the lab is actually looking at the protein itself. What does it do? How does it look? Those different structures and different forms that it forms. Can we mimic this? Can we study it in terms of how is that bad for your brain? And we actually do in this lab, and we've been very successful of discovering completely new changes in the protein that were never reported before, and we're working on this project right now. We also collaborated with other PIs at Sanders Brown when we actually show that this protein affects brain activity and your vasculature. So it kind of links with that stroke and what happens when your vessels are not working properly. The third one is just example. I'm not going into details here, but we're also looking at how it hijacks the brain energy. I mentioned that the brain uses 20% of your glucose, right? So we kind of have now a whole project looking at how this TDP-43 kind of impairs those mechanisms and what do it mean therapeutically to target it. Okay.
I'm having a little bit of post-traumatic stress from biochem here.
Okay.
But you'll forgive me for that.
Is that the TCA cycle? I know, I had it too until I had to learn it. So I mentioned that we discovered this new modification is actually called citrullination. It takes this TDP-43 protein from being aggregated like this, it makes it looking like this, like sand corns, I say. And why is that important? This is important because it's creating these all new shapes and forms of the protein that we don't even know what they do in terms of toxicity to the neurons, right? And this is work that I got support from NIH, and we're publishing hopefully very soon, and will be out there for communities, for scientific community, and obviously communities at large. But it changes. I mean, this is, again, this TDP-43 in itself, this is the new ones that we discovered. I can play the video if we have time, or people can play on their own, but you see the changes in their structure, drastic changes. And we're doing much more studies, and it's trying to understand it, what it actually does. Next is, I wanted to touch upon some of the clinical trials that we have going on at Sanders Brown. And here, we kind of, again, to reiterate, we have major focus on translation, and hence where these clinical trials really come in. We have Alzheimer prevention trials. We have START study, which is actually a study that, and I don't want to misspoke here. Actually, I'm going to start with SMART-HS first, because this is the first clinical trial in Word for late, and this is driven by Dr. Jaika and Pete Nelson. We have a study that is Harmony at Home. This is actually therapeutic and at work with the patients and caregivers to make sure that they have the right settings to help the patients. Patients with Alzheimer's disease, we have this sleepy study that actually looks at anxiety through cannabis to look at how to improve the symptoms of anxiety and agitation in Alzheimer's disease. That is a big problem, and the START study that is actually a clinical trial phase 2, and that is for amyloid beta as well. Okay. And then there can be, as we mentioned, that is actually the clinic part of it has been the home when those infusions occur. Right. So again, we're kind of moving towards our goal, enable healthy brain aging for Kentuckians, but also kind of bring a bit more precision care by looking at those different mechanisms and different pathologies in the brain, trying to see what works for different patients, as this is a very mixed and complicated pathology. And then, as I mentioned, in our laboratory, we're really looking at this mixed dementia, trying to understand how this new protein kind of affects dementia progression, and how do we tackle it? And I believe this will be the last slide of obviously my laboratory, but also, yes, I also wanted to kind of have for your clients a bit of information if they wanted to go and check more
Yeah.
George Brown and get in contact with people.
That's great. It's a great team. I'm kind of jealous of
Thank
you
you.
all because I look at some of the history in these slides. The drugs that are coming out and the bits that you've studied, and I feel like I say this like every year I come see you all, but it's like I see the progress, and I feel like we're kind of on the side of a hill that we're about to start rolling down, and you all are like in the center of the universe of the place that's going to drive change in a really meaningful way over the next 10, 15, 20 years. So it's just exciting work that you all are doing and get to be in the center of.
Oh, thank you very much. And thanks for giving me the opportunity to kind of share
Yeah.
our work.
Yeah. I mean, right in our backyard, world class.
We are.
It's just it's incredible that we have you all here, and we appreciate you.
Thanks, Cameron.
Yeah.